tried to take samples for both methods, eDNA and traditional gear, in all of those locations. So we'll go into some of the results. This is decants is in the gold, water samples is in the teal. And what we saw is that the water samples not only were picking up more unique species than the decants were able to, but in locations where they were collecting the same species, they're picking it up in a higher frequency of sites. So good examples right at the top, brown goby. We got maybe 7 detections in the decants for DNA in every single site in the water sample. And that was really the trend. There's a few exceptions like walleye, bluegill, where the decants did a little better, but really the water samples outperformed. Moving on to the comparison for traditional gear versus water sample, we really saw the same trends. Water samples were picking up more things and we're picking them up in a higher frequency. There were a few exceptions down the bottom here in the gold where traditional gear picked up a few more unique species, but not enough to be like, oh, water samples didn't do what we were thinking it was going to do. Now, just something I want to point out now, and then I will bring it up in a couple more slides, is that there is still some genetic confusion with some very closely related species. Now, like I said, we had sampled Oswego Harbor for many years at this point. We had caught in long-nosed gar in a wide variety of spots in the harbor, and we were even able to collect them in a few sites in our traditional gear. But our water samples were coming back with short-nosed gar, which we were confident is not in the area. They've never been in the area looking at any maps that we had available to us. And so we talked with the geneticists and realized that those, through metabarcoding, the genetics are so similar that a lot of the times, short-nosed gar, it is long-nosed gar, but the marker just isn't there yet to be able to differentiate the two. Another thing I wanted to look at is maybe one of these gear types doing a better job, say, in the open lake, where another one might be doing better in the river. So I did just a quick hotspot comparison with species richness, and what I saw was that, for the most part, where there was the highest species detected for all of these 3 techniques, they were all roughly in the same location. The river was where we were getting the most species for all 3, but then for decants and water samples, the harbor also had a couple of areas. But they're all overall pretty similar. So some lessons learned for this, we were very impressed with what the water samples were showing us. They picked up in total 10 unique species that the ethanol decants and the traditional gear was not able to pick up. Now, I will say that for the traditional gear, it might not have gotten a total fair shake at this. Some of the species it wasn't able to detect, like brown trout, Chinook salmon, lake sturgeon, at that time of year probably would have been in deeper, cooler waters, and we set all of 2 gill nets the whole time we were there. So it's not that they weren't there. The DNA is indicating the presence of DNA, but we didn't collect them. And we were starting to realize that our traditional gear was just simply not selective at that time for those species. And as we started thinking about invasive species, we're like, well, is our gear always going to be selective, or do we have to utilize as many methods as we can to at least decide presence or absence? When it came to the decants, they were kind of the one where we were like, sure, they are picking up some environmental extra DNA that we were kind of seeing in the last phase, but overall, pretty good at representing the larvae that were in the sample and knowing the life history of a lot of the fish that we were looking for. A lot of those were probably spawning at that time, so it's a good indicator of what larvae were in the area. Like I had mentioned, I thought it was pretty interesting, the high species detection. They were all in roughly the same areas. It's not like one technique was doing better in one spot than the others. It was all roughly the same. You had mentioned there's still some taxonomic issues that need resolution. A lot of the cyprinids, the gars, there's still some things that need to be cleared up. But tissue markers are being developed literally every day for metabarcoding and qPCR. And I think at the end of the day, it really is your question that's going to dictate what technique you're going to want to use. For us, because we want to cast that wide net, a community-wide assessment with some of these caveats is still sufficient for us. If you have a question that's more one single species specific, then maybe qPCR is the way you want to go. But looking at the results we got, we realized that adding this metabarcoding technique into our toolbox had some great potential. So just quickly, I want to go over our next steps. We have a phase 4 that we've been doing in 2023 last year, and we'll continue this year in 2024, where we're not going to be doing any dedicated AIS and larval fish work, but we are going to move forward with this water sampling. And working with the Northeast Fisheries Center and actually several other Fish and Wildlife offices around the Great Lakes, we've been working on some studies to ensure best practices and protocols when it comes to this new sampling technique, especially with the Smith root filter and filtration system. Some of those best practices that we tried to tackle in 2023 were point versus transect sampling. So is staying in one location and filtering 2 liters of water going to get you the most amount of species, or is potentially moving and hopping from pocket to pocket of DNA going to give you a better result? Same with onshore, offshore. Can we pick off offshore species in our onshore sites, or do we truly have to go offshore to be able to detect them? And then single samples versus triplicate samples. Is 2 liters going to get us what we need to see, or do we need to up it to 6 liters? So with that, I just want to give a quick thank you to everyone at the Lower Great Lakes office and the Northeast Fisheries Center. Like I said, this kind of question and answer that we were doing started in 2017. So it's been a lot of work and a lot of time to get these results, and we think we're finally in a place where we can move ahead with water sampling. With that, I'll take any questions.